Hello everyone, welcome to my presentation. Myself Shwetlana from Pharm second year. The topic I am presenting is Streptococcus pneumoniae, which is responsible for causing pneumonia. This presentation is guided by Professor Suhani Solanki. The topics that I am going to cover are morphology, scientific classification, cultural character, biochemical reaction, resistance, toxin and other virulence factors, pathogenicity, epidermacy, laboratory diagnosis, and treatment. Morphology. Streptococcus pneumoniae is gram-positive lancet-shaped bacteria. Imagine a burning candle, the flame which is having one end pointed and other rounded. This kind of shape is known as lancet shape, which is possessed by this bacteria. Initially, it was classified as Diplococcus and further reclassified as Streptococcus pneumoniae. When initially it was observed, it was named as Diplococcus because two cocci cells were observed together, which were forming the pairs. And after further studies, its name was changed and reclassified as Streptococcus pneumoniae. This is because genetic character between Diplococcus and Streptococcus were same and hence named as Streptococcus pneumoniae, which is also observed in chain. Pneumococci is non-motile, non-spore forming capsulated bacteria. The capsule is a outer side covering that is responsible for protection of the bacteria. They may be aerobic or facultative anaerobic in nature. It is normal microflora of human upper respiratory tract. Pneumococcus bacteria may be an important cause of pneumonia, otitis, sinusitis, bronchitis, meningitis and other infections like arthritis, conjunctivitis and supratentive lesions. Scientific classification is as follows. It belongs to the domain Bacteria, phylum Firmicutes, class Bacilli, order Lactobacillus, family Streptococci, genus Streptococcus, species Streptococcus pneumoniae, and it is having a binomial name Streptococcus pneumoniae. Cultural characters. Pneumococci bacteria required enriched culture media for its growth. As it is a human pathogen and lives in humans' upper respiratory tract, hence it gets much nutrient for its growth. When we culture them in laboratory, all nutrients are needed to be given where proper growth can be supported. Therefore, enriched nutrient medium is provided. They required anaerobic or facultative anaerobic conditions for their growth. The optimum temperature for its maximum growth is 37 degrees Celsius and they can grow in temperature range of 25 degree to 42 degrees Celsius. The optimum temperature here means by where most of the growth can be seen, and the optimum temperature of this bacteria is same as that of the human body. The optimum pH required is 7.8 and can grow at pH range of 6.5 to 8.3. The cocci undergo autolysis by intracellular enzyme. The autolysis of cell enhanced by SLS solution and other surfactant active agent. Here SLS means sodium lauryl sulfate solution. Looking towards its biochemical reaction, pneumococci ferment inulin and this is one of its specific characteristics, and this fermentation differentiates pneumococci from other streptococci. It also ferments many sugar, result in the production of acid and no gas is produced. Pneumococci solubilize bile constantly, so it is important point considered in the process of diagnosis. It shows catalase and oxidase test negative. Looking towards its resistance, pneumococci are heat sensitive organism. Its thermal dead point is 52 degrees Celsius for 15 minutes. It is very sensitive to heat It's sensitive to antibiotics and antiseptics also. The pneumococci strains are maintained by lyophilization or by inoculation in semi-solid blood agar. Prolonged incubation kills bacteria cell due to production of toxin peroxide. This toxin is responsible for killing the bacteria and causing the death. These strains are sensitive to many antibiotics like penicillin, amoxicillin, clavulanic acid. Nowadays, many penicillin-resistant and multi-drug-resistant pneumococci strains has become a global problem. This is because there is a production of resistance in the bacteria. Therefore, it does not remain sensitive to such kind of drugs. Pneumococci is sensitive to chemical optoxin. It is used to treat infection caused by pneumococcal infection. Next, toxins and other virulence factors. The virulence depends on capsule and toxin production, which is known as pneumolysine. The capsular polysaccharide of pneumococci is acidic and hydrophilic in nature. So, these cells are protective from phagocytosis. Pneumococci produce pneumolysine. It is membrane damaging toxin and shows cytotoxicity. Pneumococcal autolysine acts as a virulence factor involved in autolysis and contribute to virulence. Autolysis means this own death is caused by its own virulent factors, virulence effect. Now, pathogenicity. Intraperitoneal inoculation in mice and rabbit can cause a fatal infection and death can occur in 1 to 3 days. Here, intraperitoneal means abdominal region of mice and rabbit. Pneumococcus can localize in human nasopharynx and can infect middle ear and respiratory tract. These are the two regions where it is mostly affected. Generally, infection is endogenous, but highly virulent strain can also cause exogenous infection. Pneumococci are a cause of pneumonia and bronchopneumonia, and it is very common in children as the immune system of children is not completely developed. Pneumococci are associated with chronic bronchitis, which can last for very long time. Meningitis is most dangerous and serious condition in this infection. Looking towards its epidermacy, the source of spread of infection is the respiratory tract of carrier, or we can say the patient that is suffering from pneumococcal infection. The transmission can occur by inhalation of contaminated droplet of carrier. Disease result when the immunity of person is low and the person get infected with pneumococci. The physical condition of human being such as viral infection, stress, immunodeficiency, malnutrition, congestion increase the chance of this kind of infection. Looking towards its laboratory diagnosis, the sputum sample is collected from the patient. The sputum sample is homogenized and gram staining procedure is also carried out. After homogenization, it is inoculated on blood agar for about 37 degrees Celsius between a time span of 24 hours. In case of infants, serum-coated laryngeal swabs are used. Gram stain cerebrospinal fluid is used for presumptive diagnostic of meningitis and gram-positive diplococcus are observed. The capsular polysaccharide are demonstrated by using the blood, urine and cerebrospinal fluid sample. This sample undergo immune electrophoresis process. Looking towards its treatment, antibiotics can be used for treating this kind of infection that are penicillin and amoxicillin. If the strains are resistant to other antibiotics like erythromycin and tetracycline are also used. In very serious infection, vancomycin and cephalosporin drugs are of choice and can be used. Thanking you.